Okay, so I greet you wherever you are. My name is Biba Wright, and bioinformatician and a data scientist. So today we are going to look at how to make sense from a virulent factor or virulent factors that we obtain when we run aggregate or when we are able to generate or hit our genes of bacteria or any other organism or pathogen. How do we make sense or how do we visualize it using R? So follow me, and before that, please try and subscribe to the channel. And by doing that, you are supporting me. Okay, so let me share my screen, and quickly we go straight to that. So before I share my screen, let me show you this. So if I run my aggregate, this is what I am likely to get. This is the file that I have gotten. If I come to this place, what you see here, so these are the strains, the different strains of the bacteria where I have highlighted. And so you can see the sequence. Okay, so we can see the sequence identity, which is here. We can see the start, the end. And so this is where my, let me put it this way. Let me put everything in yellow, and then you see we highlighted. So these are genes, uncountable genes that you can think about. These are different genes where I have highlighted yellow. And so how do we make sense from it? How many of these genes are present? I mean, how many, what kind of sense can you make if you have this kind of a file, a CSV file? The easiest way to be able to make this kind of sense is to take this file to R and then turn it into an image. And that's exactly what I'm coming here to do. So you can see that it is a CSV file, and that's what we are going to work with. So let's get to RStudio and then see what we are going to do. Already, this is the graph we are likely to get at the end of our analysis. So you can see the strains are here, and all the genes are packed under here. So if I pick this particular gene where we can see the deep brown, it means that the genes, that particular gene, is found in multiple copies along or in all the strains. As you see the strains on the column, and then you can see, as you see the strains on the rows, and then you can see the genes which are on the columns. So this is a column. So if I pick this particular gene like a PE35, you can see how the gene behaves. And then if we want to look at the strains that has what are all the genes, we can pick a strain from here and see that, yeah, these particular genes are there, but not multiple copies, just a few of them who are there. And then you can follow it through. Where you have the deep brown, it means the genes are concentrated. Where you have light to white, it means the genes are non-existent. That particular gene is non-existent. For instance, if I pick this particular gene like #A, then I follow all the way, you can see that that particular column is practically white. So it means that particular gene is not not too much present, or it is not sparingly present along or in all the strains that we are talking about. And so that's what we are coming to do. So how do we do that? Now let's get back to RStudio and check something. So first of all, I've already written the code. So I use this particular algorithm, which I call read, read.s, and then you choose a file. So when I run that, it should take me to my local, and so you see, it's asking me to go and choose, and I'm going to choose a CSV file. So you can see that I have the most virulent MTB, that's a Mycobacterium tuberculosis, but it's a tabular form. The CSV format is here, and then I will simply pick that one. Now, let me come here and then show you something once again. If you check, the file name is actually most_this, because there are two sheets. So this is one of the sheets, and then another sheet is here. So, but I name everything under AMR, and then so these are the genes. So that's the file we are looking for. So if I go to my desktop here, if I come to my desktop like this, I am looking for the same file name. And when I get the same file name, I think most AMR, so the file is here. I simply click on that and come and click open, and then simply it will take it, it will go to R. And that's what I'm trying to do. So at the moment, I am not interested in that, so I'm going to close that app, and then because I've already run that particular line. So when it comes, we'll do our preliminary checks. And so I'm just going to check the class, because when I read it, I packed it to this variable called MTB virulent genes. So And so I'm now coming to check to see if it is present. And you can see that actually the class what is a data frame. And then we can just have a glimpse of the head. So if I fold this up, you can see this is how the data comes in, and that's how it looks like. So you can see the database, virulent factor database, which is here. So it's actually, it gives us the first 6 lines of each of them. The strain names are here. So I pulled this down a little bit. That's for the head. We can actually look at the structure, which is I don't have to waste your time on that. Now, excluding some of the columns. So if I pull this up a bit, you can see that we might not be interested in column, this column 2, column 3, column 4. So 2 to 4, we are not interested. We are going to exclude them. So what do? We say that from column 2 to column 4, that's why I said minus 2 to 4. When I do that, I am excluding those particular ones, and I pack the new output into this particular variable called mtb virulence 1. And then when I do that, if I run that, you see that I am good to go. So now if I do the head, so we will no longer see those particular ones. You see that those ones are gone. We're now left with the file. The file contains the name of the strain and then the gene, which is here. Maybe the rest too, we are not interested in them. I'm just leaving them for a moment for some reasons, but I'll take them out as well. So when we do that, what is it? So now we check the head. And so what is the next thing to do is to simply do a count. The count allows us to aggregate the genes. So each of the gene is like, these are factor analysis. So it will count the number of times that particular gene has appeared. So when I do that, if I run this particular line 341, so I have counted the genes. So it will count. And so if I do that, I can just look at the head. And then you can see, I'm not going to run all of them. So you can see that the gene is here, and then the number is around here. So if I should look at all of them, if I want to look at all of the genes, how it has aggregated all of them, I can simply just say view, but count. So if I have view, I can simply just say AGG, and then I'll run that. So you can see that all the genes and then the total number which is there, they are all here. So the genes are there, the total number is also there. So this particular gene, they are about 143. Then yeah, P5, that gene about 286, then in that order. So you see that all the genes are there. So that's what we have done there. Now let's use crosstabs to see if we can make much more sense. So crosstabs allows us to be able to check the frequencies, practically the same as what we did in the top there. And so I'm going to run the crosstabs and then look at how it looks like. So the crosstabs will simply, each particular gene, you see the total number, which is down here. So it will aggregate the gene. So when you are looking at this particular gene, there are about 144 of them inside the entire data set. And so that's what we are doing at the moment. And so we go ahead, then we sort it out. So when we are sorting it out, it allows us to pick from those with the lowest and then to the highest, to the lowest number of the gene that has a lower number of copies in the entire pan genome as compared to the gene that has the highest number of copies in the entire pan genome as well. So we use sort, and then so I'm just going to add here sort to this particular whole, and then when we do that, I decided to run it to pack it into this particular variable called most virulent. So if I run that, you can see that it will now rearrange the gene. So this particular gene, those that haven't appeared only once, you can see them here, 1, 1, 1, 1, 1, 1, all the way. So these are the most prevalent genes we have seen so far, 385. So gene ESXH is the one that has the most prevalent in the entire pan genome of Mycobacterium tuberculosis, as far as this particular data sample that I'm working with is concerned, then in that order. So we've sorted them like that, and we packed it into this particular variable called most virulent. So when we check the most virulent, what class of is actually a table. So it comes in the form of a table. Where yes, we are using X tabs. So we are using X tables. It's actually a function in R. So now we could actually write whatever that we have here into a file, so that when we go to our desktop, we can see it. For now, it is in our environment. Let's say that I wanted to just put this information just like it is on my Microsoft Word to make sense, or to now write my manuscript on it. So I have to pack it out of R. So you need to write it. And that's what I'm doing here. So I'll run the second line. So I've written it to, also there's an error here, cannot open file. Yeah, permission denied. Yeah, I need to add that. So I'm going to ignore that. That's not the focus of today's work. I'm focusing on how to visualize. So now let's try and see if we can plot what we have seen here. But it's a table. Let's try and see if it's possible to plot it. So we can just have a glimpse. So now we have already sorted it and we plotted the sort. So we can see the most prevalent or the most domineering gene is this, as we can see around here, then in that order. And so FBAB gene, which is around here, so you can see all the genes have been summarized, and now we can now see how we can see a visual impression of it. But here, the problem is that it only tells us this particular gene, this is the quantum of it that is present in the entire pangenome. But you cannot tell whether this gene is much more present in this particular strain or not if we are using this particular plot. So that is why, let's continue and see. So what do we do again? So this time around, what we are going to do, we are going to use the cross tabs again, but this time around, we are saying that we want to have the frequency, that is n, of the X file that contains the name of the strains and then the gene as well. So it will simply regroup the strains according to the number of the genes which are present in each of them. And then we'll pack it to most virulent strain. So run that. And then when I do that, come and check the class of that. And so it's a table as well. So this time around, when I decide to run this alone, look at how it looks like. Now it looks a little bit beautiful. So we can now see that we have the strain here and we have the gene there. So if I pick this particular strain, I can see all, and then I'm looking at this particular gene. It is present only once in that particular strain. This particular gene 2 is present only once. ECCA5 is present only once, then in that order. And so we continue going and then see what we can make out of it. So I could have also written this. I've already written this to, I'm writing it as a CSV file so that it goes to my, the same permission denied. Okay, so what I'm going to do, I've already done that part of the work, so I'm not going to bother you. So and I think I need to activate some package. That is why it is not. So mostly you need to read the error message. And so they say error file, if else append, and then cannot open the connection. In addition, warning, the file, okay. I'll leave that part for now, and then let's continue. So now, I have already brought it back because we already have it, which is here. So you can see the strain, and then we can see, but here there is something I did not do. I should have taken the dot faster. Dot faster is a repetition. So there is a way to be able to clean that part up, but I don't have the time. So what I can do is to simply visualize it. So now this is where the whole video is all about. Now we've seen how to be able to get to import it into our environment, how to be able to check, to regroup it. And then now let's look at how to visualize it. To be able to visualize, you need to change it into a matrix because we want to use a heat map. And to use a heat map, you need to change the entire data into a matrix. So at the moment, as it stands, if you check the class, like as we have it here, if we check the class of the most V, like I'm telling you, I've already read it into this. That is why I'm not reading it any longer. And so this is now our new variable called most V, the most virulent. So if we check, if I say the class of the most V, if you run it, it's a data frame. Yeah. So once it is a data frame, we cannot visualize it using a heat map. We need to convert it into a matrix. So so let me put this part. So visualizing the genes in heat map. So now what do we do? So we say we are converting the data frame into a heat map. So we use the function as.matrix, and so most V. But in this case, we are excluding one of the columns, the first column. The first column is, come and check the first column. You see that the first column here, the first column here is the name of the strings. And then you cannot do this if you want to convert everything into matrix with this, the first column being strings or being characters, it will not work. So you need to exclude that out. So that's why I added -1 to exclude that part as a character. And so we now, the rest, it would be left with the rest of the columns, and then we convert that into matrix and pack it into this new variable. And so that's what I have done around that, and that works just properly. So now if you check this new variable, it will be a matrix. Okay, so you can see that it's now a matrix. For instance, if I come here, if I do a class of the most V, as this variable here, if you run it, you see that they tell you that it's a matrix or array. It's a matrix array. And so that is it. So we've done that, but the column we have removed, which is the names of the strings, we need them. So we now take that particular column and assign it to become row names. So that's why we use a function called row names of this new variable we have here. And then we are using the old variable, and we are using the column name. The first column is column X. And so when we do that, we are saying that use the column X, which represents -1 that we have removed, the first column we have removed. Then use that one as the row names, and then put it here. So when we run that now, and then we go to check that variable again, now that we have run it, when we check it again, you see that even though it's a matrix, but we now see the row names which are over there. You can see the row names are now hanging around here. So the matrix is on one side, the row names are on the other side. So here we could actually use a bar plot to try and visualize it. And then let's see whether we can build a bar plot of it. But the bar plot is not making any sense because the data is huge and it's too clumsy. Let me try and see what if I can zoom it for us to see. And so it's taking time, but it will definitely. So if we do the bar plot, this is how it's going to look like. And I'm trying to, okay, it was coming and I almost. So this is how the bar plot looks like. You see? But at the same time, we still cannot make too much sense from it because the row names, the strings, the names of the strings are not there. You see? So you can only see that this particular genes or the genes which are here are much more prevalent, but that's not what we want. So the best way is to use a heat map. So we are saying a heat map of this particular variable, and we are collapsing we don't want any clustering from the column side and clustering from the row side. So that's why we say the column variable, we collapse it. That's why we assign it to NA, and the row variable, we also assign it to NA. And when we do that, we'll produce a heat map that doesn't have clustering. If not, it will get us a heat map. So we run that and then see. It takes time for heat map to build, so you need to bear with me. But it's running anyway. Yeah, so the heat map is here. So it comes, and that's how the heat map looks like. And this is how, so you can see that now we have the genes which are on the columns, and then the strains which are on the rows. So now if I want to know if this particular gene is the most prevalent, I can now see that, okay, I can trace the gene and say that, okay, the gene, even though it's most prevalent, it is not found in this particular strain, or it is primarily present in this strain. I can cross-check and see which strain is that. And it is strain DE10.faster. That's why I wanted to remove the faster. So that's how we check it. So we can see this particular gene, these two genes here, hash A and then hash C. Those genes are not, are practically absent in most of the strains, except few of them. And so we can see the place is white. So we can also see that the strains from here up to this place, if you look across, you see that they have the brown shade. So whatever you have the brown shade means much of the genes are present there. If the shade is deep brown, it means that particular corresponding gene has a lot of copies in that particular strain. And this is basically how to be able to visualize virulent genes if you have already obtained them. I hope this video helps you to be able to do this. You can do this for antibiotic resistant genes as well. Just follow the steps one after the other. Now, let me summarize, and then I'll leave you. So all we have done is that. So let me pull this to this side. I'm going to clear this, and then I'm going to move this a little bit. So all we have done is that we have our data, which is here, and these are the genes we have obtained. We need to visualize these genes because I cannot go through this whole thing and see where do I have ES, X, H, how many of them are present. It's going to be, what is it? It won't bring me any, So we need to visualize the genes. We need to turn it into an image, and the image will carry all the information that we need. So what do? We need to send this to our environment, and that's what we did. So we use this particular function to do that. After that, we do our preliminary checks. Then we went ahead to exclude some of the columns we don't need. And then we went ahead to aggregate the genes to see how, each of them, how many are they. And so when we check this, or when we just looked at the head of this, we saw it, which was sitting here. As you can see, the gene and the total number that is there. Then we decided to use cross tabs so that we can add let's say, the names of the strains. So the names of the strains occupy a column or a variable called x.file, and so plus the gene. So now it will aggregate the number of genes per strains, and then in that order. And that's what we have done, or we pack into this new variable. And so after we've done that, we decided to write that particular variable, to write it into a file in into our desktop, and we imported it again using this particular because we did that because we wanted to remove the .faster because I didn't want to write a long codes here to be able to do that. So I went to Excel to remove this, and then I imported it again. But this time around, I did not remove this from from my X, or I didn't go to my desktop, but this was the one that I have removed. So as you can see, I've already removed it, but I imported the one that has the .faster. That is why you are seeing it there. So now, after we've done that, now we come again to now do the visualization. To do the visualization, you remember, we have to convert it into a matrix. Converting to matrix, you cannot convert the numbers plus the strings, which are characters. So we excluded the column that contains the strings and the characters, and then we use those ones as our row names. That's why we said -1 here. We assigned it to use this new variable. And then we decided to use that same column that we excluded, which contains the names of the strains, as our row names. After we have done that, we now have our complete matrix data, and we use the heat map to visualize it, and we collapse the clustering, and that is why you don't have any clustering around here. So this is how to be able to visualize your genes, or either your virulent genes or antibiotic resistant genes. I hope you've learned something small. Thank you. If you enjoy my videos, why don't you subscribe to my YouTube channel, and then by doing that, you are supporting me. Thank you very much.